So it's taking forever for me to upload my videos onto this iMac so that I can do some editing. So I'm trying to use the Zoom app to do some editing. I'll give this a shot and see if this works. So remember the seeds I started for the seed viability exercise we did in the lab? So these are the radish seeds. I think I let them go a little longer than I should have to examine their roots. So as you can see, they're kind of a mess, and I was trying to isolate individual roots there. So instead, I checked on the grass seeds that I had started, and they looked a little better, but still they were a little bit of a challenge. So let's take a look at the grass seeds. So here are the grass seeds. Remember, grass seeds are monocots, and these are not as root bound as what I saw with my radish seeds. So these will work a little better. So I'm able to isolate individual seeds and their roots. But when I started looking at them with the projector, I realized the projector magnification was not high enough for me to show the details that I want. So I'm going to show those details under the microscope instead. So we're going to be describing the structure of the root tip here using the grass, which is a monocot. We're going to see similar features in dicots, and we'll talk about the internal structural differences between dicots and monocots later on. But if you look at the figure in the lab manual, it should show these characteristics that we're going to be talking about. So the root tip is covered by the root cap, and the root cap is produced by the apical meristem. And the apical meristem also produces the rest of the root here. But the root cap's function is to protect the root as the root pushes its way through the soil. And it constantly has to be regenerated because those cells are getting sloughed off. You can see some of the cells sloughing off here on the root cap. Then we have our apical meristem, which is the region that contains those initial cells that divide relatively rarely. They give rise to derivatives. And the derivatives divide many, many times, and that region is known as the area of cell division. So just behind the root apical meristem, which you really can't make out here. We'll talk more about the root apical meristem later on. But there's the region of cell division. So this is going to result in many, many, many cells that are then going to begin to elongate. And that's going to lead us to this region here called the area of cell elongation. So most of the growth, if you consider the elongation being growth, which it is going to occur there. This is going to push the growing tip of the root through the soil. Behind the area of cell elongation is a region known as the area of cell differentiation, and this is where the cells begin to differentiate, also forming all these root hairs. So these root hairs are extensions from the outer epidermis of the plant, and they are going to be involved in nutrient uptake. So water and various types of minerals will be taken up in this region here. These root hairs can only form in this area following cell elongation, because if they were found in the area of cell elongation, they could be sloughed off as the root pushes through the soil. So this little video is just going to show a movement up the root here, showing the region of cell differentiation, all the root hairs that are produced. So lots of root hairs. So this area is quite extensive. And in a root that's in the soil, these root hairs probably wouldn't be so visible and extensive. Oh, pause this for a second. And that's because these root hairs only last so long, usually about 2 weeks, and then they essentially wither away and die, and that just forms the mature root. And the mature root, its epidermis is highly waxed so as to prevent any water that's being taken up from being lost. And you can see this other root over here. This is a monocot, and monocots do have branching roots. But this is not a branch root. This is probably one of the roots that are forming from what's called advantageous root growth. So the roots of monocots tend to be more fibrous than in dicots, which have more of a taproot system. And so this is just the beginning of the many, many, many roots or advantageous roots that are going to be formed as the plant develops. But let's follow the rest of the root here. So this is an area where the majority of the root hairs have fallen off already. And that's about the extent of it before I think it's too big. Notice all the little swimmers swimming by. Okay, here we have the same grass root at a total magnification of 100x. And you can see down here at the root cap, the cells sloughing off. And this little darker brown region is the root cap. And you can sort of make out some of the cells there. So let's just kind of follow along the root a little bit. The reason why it's not centered is because the little program I'm using to capture the video off the microscope, for whatever reason, moves it from the center of the field of view. But there you can see some of the root hairs. And I think I pause it here in a second. You can see how they're just attached or part of the epidermis. You can also see some little swimmers swimming around. Those are most likely different types of protists. And I never kind of understand how they show up, but they're very good at showing up and occupying various habitats, including the paper towel that I was growing the grass on. But you can see the root hairs coming out from the epidermis. This increases the surface area for the absorption of water nutrients there in the root. We'll talk a little bit more about their role and how nutrients are taken up by plant roots when we talk about plant nutrition and soils later in the quarter. Oh, there's another little swimmer. Cells are not static dead things, so while I was looking around at the roots, I noticed all this beautiful cytoplasmic streaming going on. So this is movement inside of these root hair cells, and this is caused by, well, it's caused by various types of cytoskeletal components that literally walk the various organelles and structures inside of the cell along the cytoplasm of the cells. So you can kind of see that movement here in this slide. So this is a total magnification of 400x, and you can see, let me pause that for a second. Let me pause. Down at the base of the root here, where it attaches to the epidermis, you can see, that this is part of the epidermis, and you can see that the cytoplasmic streaming goes into the cell that actually gives rise to that whole roots here, because it's all just one cell attached there. Cytoplasmic streaming doesn't connect cell to cell, though. So it's only going to be isolated to that one cell. You can kind of make out the boundary of that cell there. But you can see all the cells around it also go through the process of cytoplasmic streaming. See, it's kind of moving around in those cells. Very cool. I know this doesn't have anything to do with the topic of the root, but kind of fun stuff nonetheless. So this is the root from the radish. So this is a dicot root. Again, you can make out the root cap. It's a bit of a mess because there's just so many roots kind of overlapping one another here, because the root from the radish is kind of older and has been branching quite a bit. So actually I want to show it because I want to show the point in which a branch root is occurring. So remember, in stems, branches form specific regions called nodes, and these regions between nodes are internodes. This is not what happens in roots. In roots, the branch roots or lateral roots form wherever the root decides to put them, and this is dependent upon what kinds of nutrients that are being sensed by the root. So let's see what it looks like. Oh, by the way, this is a magnification of 100x total. So here we have the root cap. You can kind of make out the root hairs along this root. This is a lateral branching root. And below there, you can see the larger root that it's branching out of. And when branch roots or lateral roots form, they actually form from the inside of the root and push their way out. Like the analogy of the Alien movie, so that it actually pushes its way out and leaves a kind of hole in the main root. Here it is at lower magnification. You can see that little branch root coming out from the main root. But otherwise, there are similar structures as what I was showing you for the grass root, similar sorts of structures of the root tip.